Hello and welcome to lesson number 36, and this lesson is entitled Process Validation. The objectives are as follows: a brief introduction to process validation, some definitions around process validation. We will discuss the advantages of validating your processes and the different approaches that you can take. We'll talk about the types of validation as well as the some of the basic things that need to be included in validation protocols. Revalidation is discussed along with documentation requirements. So the definition from the FDA is of process validation: establishing documented evidence which provides a high degree of assurance that a specific process will consistently produce a product meeting its predetermined specifications and quality attributes. And just to discuss validation then, it means confirmation by examination and provision of objective evidence that the particular requirements for a specific intended use can be consistently fulfilled. And I've included a CFR reference there. Some validation examples would be process validation, cleaning validation, sterilization, test method validation, or software validation. Verification means confirmation by examination and provision of objective evidence that specified requirements have been fulfilled. Examples include verification, design verification, and process verification. So to continue with the definitions: process performance qualification, establishing confidence that the process is effective and reproducible. Product performance qualification, establishing confidence through appropriate testing that the finished product produced by a specific process meets all release requirements for functionality and safety. And finally, prospective validation, validation conducted prior to the distribution of either a new product or product made under a revised manufacturing process, where the revisions may affect the product's characteristics. So some of the advantages of process validation, or ensuring control over your process, includes things like reduction in rejection and rework, reduction in downtime, improving efficiency and productivity by optimization of your process, easier scale up from development work, improved employee awareness of process, reduced testing in process and in finished products, compliance to meet regulatory requirements. The following is a flowchart or a sequence to validation, or how it happens. The first thing being your process is developed, normally at lab scale, so in your fume hood. It's then optimized through the laboratory, so the process is improved further through lab testing. Validation of sub-processes, which is normally things like validation of your analytical methods, ensuring that your equipment and your utilities are qualified, the cleaning processes and the methods are validated, and all your instrumentation in your laboratory are qualified and validated. Optimized plant or batch is the next step, where your process is carried out in the actual plant. So they're called optimization batches, or practice runs, if you like. And following that, we then go into process validation. That's normally carried out on 3 consecutive batches in the plant at plant scale. And finally, we consider your process to be continually monitored throughout its life cycle. So we talk about ongoing process validation, where you're constantly looking to make sure you're in control of that process. The different stages involved in process validation would be, firstly, your process design. So the commercial manufacturing process is defined during your design stage. It's based on knowledge gained through your development and your scale-up activities. Stage 2 then is your process qualification. During this stage, the process design is evaluated to determine if the process is capable of reproducing commercial manufactured material. And finally, stage 3, continued process verification. So ongoing assurance is gained through routine production that the process remains in a state of control. And that's very important to ensure that your process is continually monitored throughout its life cycle. So again, just to talk a bit about purpose of validating your processes, and really it's to gain a high degree of assurance in the performance of the manufacturing process, such that it will consistently produce API and drug products meeting those attributes related to the quality, efficacy, strength and purity that you've claimed. It's to confirm the process and the manufacturers remain in a state of control over the life of the process, even as materials, equipment, or the environment, personnel, or the manufacturing process changes over time. Manufacturers should use ongoing programs to collect and analyze product and process data to evaluate the state of control of the process. So the 3 the different types of process validations are listed here. The first one is prospective process validation. It would normally be expected that process validation be completed prior to the manufacture of finished product that's intended for commercial sale. So prospective validation is normally where a new formula must be validated before routine production commences. Concurrent validation, this is where this is not possible. It may be necessary to validate processes during routine production. Retrospective validation, processes which have been in use for some time should also be validated, but these are processes that are well established, and you can use data coming out of those processes for retrospective validation. And then, of course, revalidation needs to be completed if your original process changes in any way. So just to reiterate further on the prospective validation, prospective validation involves manufacturing, sampling and testing of validation batches according to a pre-approved validation protocol, and we'll go into further detail on what should be included in a validation protocol in the next few slides. Validation should be conducted prior to distribution of your new product, or if there's changes to the existing process, you need to carry out prospective process validation. Prospective validation is established, documented evidence prior to release of your product based on a pre-planned or pre-approved protocol. It's conducted prior to distribution of either a new product or a product made under a modified production process where modifications are significant and may impact the product characteristics. It's a pre-planned scientific approach and includes initial stages of equipment validation. The concurrent validation, it's established, documented evidence that a process does what it's supposed to do based on information generated during actual implementation of the process. It may be practical under certain circumstances when the number of batches are limited, when a previously validated process is being transferred to a third party, for example, or to another manufacturing site. Retrospective validation is established, documented evidence that a system does what it's supposed to do based on the review or analysis of historical data. It has become synonymous with achieving validation by documenting all historical information, which is normally, say, 20 to 30 batches are considered to be enough information to establish retrospective validation. And finally, revalidation. Well, this is nothing short really of repetition of the whole validation process or a specific portion of it, and it's necessary in some situations where changes may be in some of your key starting materials, or if there's changes in the raw materials or process parameters, for example, mixing time, drying time, even batch sizes. An obvious one would be a change in the plant facility or some of the equipment used, and sometimes changes in the final specification of the API may require revalidation of your process to ensure that it's capable of falling within that predefined specification limit. So your validation protocol then is a pre-approved document that needs to be approved by quality assurance being the main sign-off, and some of the things that needs to be included within that protocol will be the purpose and scope of the study, along with the type of validation. So is it a retrospective, concurrent, revalidation, and the justification for carrying that out, a list of the responsibilities, batch selection and criteria, so what batches are you going to do the, if it's a validation, you pick 3 batches, what batches give the numbers, your process description and flow, along with your batch manufacturing instructions or your BMR. Some of the key equipment should be listed, as well as the raw materials or the key starting materials. The CPPs, or the critical process parameters, as well as the CQAs, need to be identified in your protocol. Your sampling plan, so how or who or when samples are taken, along with the acceptance criteria of those samples taken. How will deviations be handled, and the approvals, of course, which include the key personnel, whether that be regulatory, production, QA, etc. The sign-offs are normally included on the first page of the protocol. So some important validation considerations that you need to bear in mind are listed here. The first one is in-process controls, and these are to ensure that the final drug product meets its quality requirements and how you control batch-to-batch variability. You need to establish levels and frequency of routine sampling and monitoring during your process. So these are termed in-process controls or in-process samples. And you need to define specifications and acceptance criteria around those samples. The test methods associated with those in-process samples needs to be fully validated before you can run the samples. The sampling plan then is the plan for each unit operation. So the sampling points need to be listed along with the number of samples and the frequency the sample is taken. So where in the process stream it's taken, the timing around that or when it's taken, how the sample is taken, along with the quantities that are taken. A selection of your sampling points can be based on risk analysis as it relates to the particular attribute under examination. Something to be aware of here would be your PAT technology, and can you use online or at-line techniques to carry out your sampling of the process stream. Critical process steps or parameters. So what parameters have impact on the product quality? For example, temperature, agitation speed, pH or time, holding time in a bulk tank. You need to establish the manufacturing conditions based on scientific judgment and cumulative data that was taken from your design of experiment or your pilot runs on your pre-commercial batches. Define and monitor the critical process parameters and the CQAs. Define the acceptance range or the specification limits for these key process steps. Some of the things to be aware of would be, say, your drying time, your granulation time, your tablet compression, because that might change the crystallization form. So you need to understand your process in order to determine your CPPs and your CQAs, and to define your acceptance criteria around those CPPs as well. So the acceptance criteria to be considered as part of your protocol, you need to take into account the product quality along with the yield, the CPPs and the CQAs to determine your in-process results. Continued process verification. So the goal is continual assurance that the process remains in a state of control during commercial manufacture. Evaluating the performance of the process identifies problems, and CAPAs, corrective and preventative actions, are implemented to ensure that the process remains in control. An ongoing program to collect and analyze product and process data that relates to product quality must be established. The data collected should include relevant process trends and quality of incoming materials or components. It should include in-process controls and finished product testing. And also you need to consider the equipment, the facilities used throughout your process. Some references here, the ICH guideline documents along with the United States FDA guidance documents, and there's another one here on the EMA guidance documents as well. They'll all be available through the supplementary material link that will be included on the links. So please take a read through those when you can. And that's the last slide. Thanks for listening.